Hello, dear students. I welcome you all on behalf of Sri Jendra Rank Zone classes. Dear students, we are in a continuation of the chapter that is photosynthesis. And in this lecture, we are going to talk about the C4 pathway and CHEM pathway. First, we will discuss about the C4 pathway. So, as we have discussed in the previous lecture also, that when the first stable product after CO2 friction is of 3 carbon, we call it as a C3 pathway. And the first stable product after the CO2 friction, if it is of 4 carbon, we call it as a C4 pathway. And here, this is going to be the 4 carbon first stable product. So if you talk about C4 pathways, C4 plants are special. In those plants where the C4 pathway takes place, they are known as C4 plants. And it is known that C4 plants are special because they have a special type of leaf anatomy. And this special type of leaf anatomy that help a C4 plant to tolerate high temperature. They give them a capability to show a response to high light intensity. Lack of process called a photorespiration, that is a C2 cycle, which is absent, which is present in C3 plant, but it is absent in C4 plant, and they have a greater productivity of biomass compared to C3 plant. So these all things are the comparatively better in C4 plant. That is due to the special type of anatomy. This is present in this C4 plants that is known as cranch anatomy. And in the cranch anatomy, they have a particularly large cells around the vascular bundle of the C4 plant called a bundle cells, and the leaves which have such anatomy is called a cranch anatomy. So they have a special type of anatomy that is known as cranch anatomy. So cranch means what? Cranch means a wreath. Wreath, that is a reflection of the arrangement of cells, means if one cells are covering the other in a circular manner, then we call it a wreath, means wrapping. Okay. So here wreath means what? The cranch anatomy. In cranch anatomy, in this, what is happening? The vascular bundles means xylem and phloem are covered by a bundle sheath cells, and these bundle sheath cells are large size cells around the vascular bundles. And the bundle sheath cells are characterized by a large number of chloroplasts. They have a thick wall which are impervious or impermeable to gaseous exchange, and no intercellular spaces, and they do not have intercellular spaces. So if we draw it properly, that how this cranch anatomy will look like, suppose, then we can draw like that. This is a suppose xylem and phloem. These are the vascular bundle. Surrounding to that, you will see the presence of layers of bundle cells, and these layers of bundle cell are present surrounding to this. So this is vascular bundle, and these are bundle sheath cells. Now surrounding to bundle sheath cells, we will see that surrounding to bundle sheath cell, you will see that there is a presence of mesophyll cell. So this concentric arrangement of this bundle sheath cells around the vascular bundle is known as cranch anatomy. So if we talk about some more features of this cranch anatomy, then we can write some features of cranch anatomy. And in the features of cranch anatomy, you will see that here the mesophyll cell is not differentiated into palisade and mesophyll cell. Mesophyll cell are not differentiated into palisade and spongy tissue. Into palisade and spongy tissue, that is a spongy mesophil. Okay, so spongy tissue is palisade parenchyma or spongy parenchyma are there. Okay, which are there, a mesophil cell differentiated into palisade and spongy parenchyma or spongy tissues or spongy mesophil and palisade mesophil in the dicot leaf or dorsiventral leaf, but not in case of the isobilateral or monocot leaf. And such thing is seen in case of this cranch anatomy. Then you will see that cells of bundle sheath cells are arranged in a concentric rings. Okay, bundle sheath cells are arranged in concentric rings, concentric rings around vascular bundles in a rat manner. And these cells of mesophil and bundle sheath cells. So it is known that mesophil cells and mesophil cell and bundle sheath cells are interconnected. They are interconnected by plasmodesmata. And we know that plasmodesmata are the cytoplasmic connection. So these mesophil cell and bundle sheath cells of this cranch anatomy are interconnected by plasmodesmata, and we know that plasmodesmata is a cytoplasmic connection between adjacent cells. Suppose this is the mesophil cell and this is the bundle sheath cell. So these are connected by this plasmodesmata. Okay, so that is the thing. Apart from this, you will see that bundle sheath cells are arranged in several layers around the vascular bundle, and as we have discussed in the previous slide, they have a large number of chloroplasts. They have bundle sheath cells have a thick walls which are impermeable to gaseous exchange, not water, only gaseous exchange, and there is no intercellular spaces. Okay. Now this is the cranch anatomy. Apart from this, you will see that these C4 plants have a dimorphic chloroplast. C4 plants have dimorphic chloroplast. Now this dimorphic chloroplast, if we talk, this means that the chloroplast of mesophil cell and in dimorphic chloroplast, the chloroplast of mesophil cell and bundle sheath cells are different, and it is known that the chloroplast of mesophil cell are small in size. The chloroplast of mesophil cells are small in size and granule. Granule means with grana. Means thylakoids are arranged in form of granum. These are the thylakoids. And these thylakoids are arranged in granum. So we call it as a granule chloroplast. Now, if they have a granum-like arrangement, means they have what a press and non-press region, means they have both PS1 and PS2, and if both PS1 and PS2 are present, means the chloroplast of mesophil cell can perform a cyclic as well as non-cyclic photophosphorylation. And if they are able to perform both cyclic and non-cyclic photophosphorylation, they it synthesize both ATP and NADPS2. Whereas if we talk about the chloroplast of bundle sheath cells, the chloroplast of bundle sheath cells, if we talk, they are large in size are large in size and agranar, means that is without grana arrangement. Means in chloroplast, if you see, these thylakoids are not present in grana, they are scattered. So without grana, if they are okay, so these are the agranar, or these are the thylakoids which are scattered, which are not arranged over each other, and they are not forming a grana. So granum is absent, means they have only free surface, they have only unoppressed region. So due to unoppressed region, they have only PS1. And if they have only PS1, they perform only what? A cyclic photophosphorylation, and they carry out only ATP synthesis. So they have only PS1, so only ATP synthesis occur. Whereas here both PS1 and PS2 are there, so both ATP and NADPH2 are synthesized. Okay, so this way the diametric chloroplasts are there. Now, apart from this, understanding this, it is also known that this C4 plant which have this characteristic, and now we have to understand the mechanism of this C4 plant. But before that, we remember this pathway was discovered by H. and Slack. So C4 pathway is also known as H and Slack pathway, or dicarboxylic acid pathway, or cooperative pathway. Means here if we will study when the pathway, we will understand why they are known as such. Names are given. And this C4 pathway is occur in the C4 plants. So C4 plants which carry out C4 pathway, they are known to adapted dry tropical regions. And this C4 plants, if we take the example, then examples of C4 plant include sugarcane, maize, sorghum, amaranthus, atriplex, etc. Okay, these all particular examples are there in which you will see the C4 C4 plants are there. Okay, so now if we talk about the mechanism of the C4 pathway, this is going to take place like this. So let's discuss the C4 pathway, how it takes place. So first we have to draw the cells. So this is the thing in which you will see this is the cell wall of plant cells, and this is plasma membrane. Such a way these are arranged. Now what reactions take place that we have to understand. Let's discuss the reactions. So here this is the cell wall, this is the plasma membrane, and this is mesophyll cell. Actually this one is mesophyll cell and this is bundle sheath cell. And here this is vascular bundle. Okay, so let's draw this vascular bundle somewhat bigger. Okay, so this way these three are arranged. Now what happens, first of all, the atmospheric CO2 gets inside the mesophyll cell, or it reaches the mesophyll cell, and this CO2 converted into SCO3 minus, and this SCO3 minus react with PEP, that is phosphoenol pyruvate, which is of 3 carbon. Okay, so what is PEP? PEP is phosphoenol pyruvate and it is of 3 carbon. Okay, now when PEP react with SCO3 minus, the fixation will occur and it is going to form a OAA, that is oxaloacetic acid, which is a C4 acid. It is of 4 carbon. Okay, so OAA, it is oxaloacetic acid and it is of 4 carbon, because of we call it as a C4 acid. Okay, now after this, here oxaloacetic acid with the use of NADPH to NADP converted into malic or aspartic acid, and these are also of 4 carbons, so we call it as a C4 acid. Now this malic acid or aspartic acid passes or transport occur of this malic acid or aspartic acid to the bundle sheath cell. Now here malic or aspartic acid undergo decarboxylation, and with this decarboxylation this 4 carbon acid can converted into the 3 carbon acid, that is pyruvic acid. So this pyruvic acid is of 4 carbon, sorry 3 carbon. Okay, now further here, along with formation of 3 carbon acid, the CO2 is released. Now this CO2 undergo here C3 cycle, and with this C3 cycle it is going to form a glucose. That glucose, after conversion into sucrose, that is transported into the different tissues. Now here also, along with this NADP get converted into NADPH. This pyruvic acid again come back through this plasmodesmata to the mesophyll cell. Here pyruvic acid is obviously 3 carbon, so we can call it as a C3 acid. This pyruvic acid converted into PEP with the use of 2-ATP, and this 2-ATP get converted into 2-AMP, that is adenosine monophosphate from the triphosphate, and these 2 phosphate are used, the phosphate are used in here for the what PP regeneration. Now here the enzyme which is used, that is PPDK. Okay, so PPDK stands for pyruvate phosphate dikinase. Pyruvate phosphate dikinase. And this pyruvate phosphate dikinase, that is 15-DKB, convert pyruvic acid into the PP by the use of 2-ATP, is a low temperature sensitive enzyme. It is low temperature sensitive enzyme, means at low temperature it will denature or it will not work. Okay. Thus, C4 plants show a poor rate of photosynthesis at low temperature, and they are active at a high temperature. Because of this, they are found in a tropical climates. Okay, so this is PPDK. So this is the what whole C4 pathway. Now in this C4 pathway, what we understood that for 1 CO2 fixation, 2 ATP additional or extra are used along with the C3 cycle. Okay, so here if we talk, then in C4 pathway or C4 plants, for 1 CO2 fixation, 3 ATP of C3 cycle and 2 additional ATP is total 5 ATP, and 2 NADPH2 of C3 cycle are used. So for 1 CO2 fixation it is occurring. So for 6 CO2 fixation or 1 glucose synthesis, this 6 we multiply with 6, then 5 6 are 30 ATP and 6 to the 12 NADP S2 are used. So here 5 ATP for 6 ATP. So 2 ATP are additionally used along compared to the C3 cycle. So for 6 CO2 fixation for 1 glucose, additionally 12 ATP are used compared to the 18 ATP which are used in the C3 cycle. So additional 12 ATP are used compared to C3 cycle in C4 plants. Okay, so that is the what we can say the particularly calculation. Now here, this is the C4 cycle. Now if we talk about this C4 cycle is understood. Now if we talk about the plants in which this C4 cycle take place, it is known that C4 plants tolerate high temperature. And they work at a high temperature, so they are found in tropical climates. They show response to high light intensity. Okay, they lack photorespiration. Photorespiration is absent in C4 plants. Okay, and due to the lack of photorespiration, C4 plants have greater productivity. Of biomass. Okay, so this is the what thing with respect to the C4 plant. So this is the characteristic of the C4 plant. Okay, now it is known that C4 plants are comparatively more efficient than the C3 plant. So if you understand that, then we remember that it is known that the evolution of C4 photosynthetic system evolution of C4 photosynthetic system is probably one of the strategy one of the strategy to maximize the availability of CO2 while minimizing water loss. So it is with reducing the water loss, the availability of CO2 and the fixation is maximized by this particular mud. Okay. Now, apart from this, in the pathway, let's see pathway. In this pathway, you can see here that this PP react with an HCO3. Here, peptide enzyme is used. That is PP carboxylase. So it acts as only carboxylase. So CO2 fixation will occur. And this is a bundle cell whose particular walls are impermeable to gases. So CO2 cannot come directly inside. So the CO2 will be released from here. So CO2 is accumulating around the rubisco, which is present in bundle cell. Because of this, we are this in here only carboxylation is occurring and oxygenation is not occurring, and due to which the photorespiration is absent. Okay, so this step maximizing the availability of the CO2. So it is one of the strategy, and it is known that C4 plants are are twice as efficient as C3 plants in terms of fixing carbon that is making sugar. C4 plants, it is known that C4 plants losses only half as much water, only half as much water as C3 plants for the same amount of CO2 fixed. Okay, so C4 plants losses half amount of water compared to C3 plant for the same amount of CO2 fixed. Okay, so because of this, it is considered that C4 plants are more better efficient compared to the C3 plant. Now, in the pathway, the enzyme we have used, that is a peptase, which is also known as PP, that is phosphoenol pyruvate carboxylase. And once you will find this one of the important question they sometimes ask from this, that it is present in cytoplasm of mesophyll cell, be remember. And one more thing, in mesophyll cell, rubisco is absent. Because of this, peptase is working for the CO2 fixation. Okay, so this is the thing for the what we can say the peptase. Now we need to discuss one more important point. Now we have to discuss one more important point. So it is known that photosynthesis in C4 plants is relatively less limited, by atmospheric less limited by atmospheric CO2 levels because CO2 effectively pumped into bundle shell cells. So here, so photosynthesis C4 part is really less limited by atmospheric CO2 because effective pumping of CO2 in bundle cells occur while the decarboxylation of the malic or aspartic acid and synthesis of C4 cycle. So the CO2 concentration is increases in the bundle cell and Rubisco will act as a carboxylase. Therefore, there is little or no chance of photorespiration. And second, CO2 is not limiting factor or deficient factor for C4 plant. That is the reason. Okay. So that is all about the what C4 pathway and C4 plants. So Hatch's like pathway, what we have understood. Let us study the pathway by listing the following steps. Some of the important points again we have to note down. Primary CO2 acceptor in C3 carbon is phosphoryl pyruvate, which accepts CO2 in form what? SCO in the form of SCO3 minus and present in the mesophyll cell and react and form the oxaloacetic acid. So the enzyme responsible for this PP carboxylase peptidase. It is important to register that mesophyll cells like Rubisco enzyme and C4 acid oxaloacetic acid formed in the mesophyll cell. This is the NCRT diagram. Sometimes you will see the questions in Neet that is from this, but based on the labeling. This is a mesophyll cell, this is a bundle cell. Labeling you should remember. This is atmospheric CO2 react with phosphoryl pyruvate fixation and form a C4 acid. What are the C4 acid? Oxaloacetic acid. Then its transport occurs. Sometimes they will ask like this is A, this is B, this is C, and this is D. What are they? That you should remember. Okay, so transport occurs. C4 acid undergo decarboxylation. C3 cycle will occur. Then transport again occur, and again PP will result at it. Okay, so this labeling you should remember for this actions like pathway. Pepcase, ultimately we have understood that form of 4 carbon compound that is malic aspartic acid in mesophyll cell that transported to bundle sheath cell. Bundle sheath cell C4 acids are broken to release 3 carbon compound and CO2 are released. CO2 3 carbon compound transported back to the mesophyll and where it converted to PP again by the completing the cycle. The CO2 released in the bundle sheath cell general C3 pathway that is Calvin cycle which is pathway common to all plants. So in Butterfield cell, which is rich in enzyme ribulose 1,5 phosphate but lack pepcase. Okay. So thus basic pathway that result in a formation of sugar in Calvin cycle is the Calvin pathway. It's common to C3 and C4 plants. So C4 plants have C4 plus cycle, but along with that they have a C3 cycle. But C3 plants have only exclusively C3 cycle. That is the thing that we have understood here. Okay. So that is all about what we can say the C3 pathway, C4 pathway. Now, after this, we are going to talk about the chem pathway. Chem pathway, chem cycle, it is a cressolation acid metabolism. It is somewhat similar to the C4 pathway in the reaction mechanism. Okay. How it is similar that we will discuss here one by one. Okay. So, the chem pathway. Chem stand for cressolation acid metabolism. Cressolation acid metabolism, the term is given because this pathway was discovered in the Cressolacea family. Because of this, we call it as a cressolation acid metabolism. And this cressolation acid metabolism chemical is alternative to the C3 and C4 pathway. It is alternative pathway to the C3 and C4 pathway of CO2 fixation. Okay. And found in the plants in a dry and hot climate. Okay. If we talk about the chem plants, it is seen the plants like cacti. Cressolacea is one of the one family for such plant. And in this pathway occurs. So chem pathway is similar to C4 pathway in that CO2 is trapped by the highly efficient carboxylase. All these are that, but we have to remember some important point that the chem pathway was discovered discovered by O'Leary and Rohani. Okay, and it is observed that CO2 fixation occur during night in these members. Okay, we will discuss that how it occurs during the night. If we talk about the chem plants, we have to understand first the examples. So if we talk about the chem plants example, then that is the Opuntia, agave, pineapple, pineapple, calonche, bryophyllum, aloe vera, euphorbia, velvicia. This is the gymnosperm. Okay, all these are the stem plants. Now, if we see these examples, the all these examples are the what we can say they are the water storing plants. They are a xerophyte, but water storing xerophyte. So it is known that chem plants are succulent xerophytes. Chem plants are succulent xerophyte, and if they are succulent xerophyte, they have water conservation mechanism, or they have water conservation mechanism, so they are adapted to the areas of water conservation. So this is the what a water conservation mechanism is seen in case of these chem plants. Now they have they have scotoactive stomata. Now it is known that scotoactive stomata are stomata which open during night, and remain closed during daytime, and remain closed during daytime. Okay, so that is the thing which is seen in case of this scotoactive stomata. Okay, now here this cotoactive stomata which are open during night, and they help in particular CO2 fixation. So now how the CO2 fixation occur, that we can understand by the pathway. And if we discuss the pathway, this whole pathway, chem pathway, takes place in a one type of cell, that is a mesophyll cell. So if I draw this cell, this cell is the mesophyll cell, and this is the vascular bundle. So this is vascular bundle. This is mesophyll cell. Okay. Now in this, some reaction occur during night and some reaction occur during the day. Now we remember here I am drawing this with the dash. This is the whole mesophyll cell. But here they have open stomata during night. So this reaction takes place during night, and this reaction takes place during day. So they have closed stomata during day. And these are the open stomata. So through open stomata during night, the CO2 enters. And when CO2 enters within the cell, this CO2 converted into SCO3 minus, and this SCO3 minus react with PEP, that is phosphoenol pyruvate, in the presence of peptidase, and they form oxaloacetic acid. This oxaloacetic acid converted into malic acid, which is 4 carbon acid, 4 carbon compound. This malic acid gets stored in vacuole. As the day occurs, this malic acid comes out of this vacuole, and this malic acid gets broken up into the pyruvate or pyruvic acid. This pyruvic acid is of 3 carbon, as we have studied. Malic acid is of 4 carbon. This CO2 is getting released. This CO2 undergoes C3 cycle, and glucose synthesis will occur, and this glucose get transported in the form of sucrose. Then further storage occur in case of the starch. This pyruvic acid or pyruvate further converted into PEP here only by the utilization of 2 ATP into the 2 AMP, and this PEP goes here, and this PP get involved here. Okay, now this PP again used for this particular step. So this way 2 additional ATP are here also used, and here the reaction occurring only one type of cell, but during night the CO2 get opened, and during day the CO2 get released and C3 cycle occur. So you can see here malic acid formation occur during night, so acidification is occurring during night, and malic acid will get broken up, that is a deacidification occur during day. So ultimately the this thing take place. So here also for one CO2 fixation, 5 ATP and 2 NADPH2 are used, but for 6 CO2 or 1 glucose synthesis, 30 ATP and 12 NADPH2 are used, because pathway is similar to the C4 plant. Okay, so this is the thing which is occurring here. Now ultimately we have understood here that whole pathway is occurring, and first oxalatic acetic acid is the first product. So we have understood that here additional 12 ATP are used for 1 glucose synthesis. Here we remember peptidase and rubisco, if we talk, both are present in mesophyll cell. Okay, so this is the thing which occurred during this particular whole pathway, and that is what about the, we can say, the chem pathway and the chem plants. So here what we have understood, however, instead of having a two kind of photosynthetic cells, chem plants have only one kind of photosynthetic cells. In C4 they have two types of cells, in which the CO2 fixation occur during night and used for the making sugar during the day. In C4 plants, CO2 fixation and Kelvin cycle are separated in space, but while in chem plants they are separated in time. This is the important point. We remember that C4 plants in CO2 fixation and Kelvin cycle separated in space, while in chem plants they are separated in time. So if we talk about the MCQs, dimorphic chloroplasts are present in case of the C4 plant, and maize and sorghum are the C4 plants. The source of CO2 fixation during a Kelvin cycle in C4 plant is, that is malic acid. Malic acid decarboxylation occur and CO2 are provided. Okay, so that is all about the what C4 and chem pathway. In previous class we have already discussed about the C3 cycle, and that is all the pathway C3, C4 and chem pathway of dark reaction are over. Okay. Now we are just remaining with the C2 cycle or photorespiration which occur in case of the C3 plant. So we will discuss that photorespiration in the next class and along with the remaining factors affecting the photosynthesis. Okay, I hope you understood well. Till that, thank you everyone.